All right, so in this chapter, we're going to be looking at blood and the functions that blood plays in the body. And so we'll start with some introductory words and then begin to talk about the first of the formed elements, the red blood cells. Okay, so functions of blood are numerous. It's certainly a very important body fluid. And it is the primary functions include transporting materials. So that's the biggest thing that we do with our blood. And there's a whole bunch of other stuff that gets transported in blood, including the nutrients. So as we do digestion, the digested materials that have been pulled from the intestine and into the blood are then transported from the intestinal areas off to the liver, and then from the liver on throughout the body to make sure that every cell has its nutrient requirements met. There is also a fair bit of gaseous materials that are transported in the blood. The two most important gases, I'm sorry about that, there we are. The two most important gases are oxygen, which is required in order to make ATP, energy, from glucose, and then the waste material that's produced from that synthesis of ATP, carbon dioxide. There are lots of other gases in the blood too, but most of them don't have any biological function. So the two biggest or most important ones to be concerned with are oxygen and carbon dioxide. Also, cells produce a lot of wastes as they metabolize proteins, carbohydrates, and other materials. And so those nitrogenous wastes that are made as proteins and nucleic acids are digested. They're carried away in the blood and other waste materials as well. And then finally, it's a transport mechanism for hormones. So when we get back to Chapter 13 and have a chance to discuss the hormones that are and the endocrine system, then we'll talk a lot more about the transport of hormones through the body using blood. It's also important in maintaining homeostasis in the interstitial fluids, the tissue fluids. So the fluid in the blood, the plasma, the water in the plasma, moves out of capillaries and out into the interstitial fluids and balances a lot of the characteristics of those interstitial fluids, making them more like the blood. It's the blood serves as a mechanism for distributing heat. We see this in when we begin to exercise, and as we begin to get hot or warm up, then the skin becomes very flushed, and if you have dark skin, it becomes darker. If you have pale skin, it can become redder because the capillaries there at the surface of the skin are dilating to bring heat from the core of the body and from the muscles where a lot of that heat's being generated, and in order to then release it out into the environment at the skin. And then finally, the blood has a very important role in protecting the body from disease and from dangerous elements that may be introduced into the body. We're not going to talk very much about that at all in this chapter. We're going to hold on to that until we get to the lymphatic system, which is going to be 2 chapters from now. So I'm not going to say a whole lot more about that. So those are the primary functions that blood serves. Come on. So blood is a connective tissue. So if you think back to last semester, Chapter 5, where we learned about the tissues of the body, we described blood as a connective tissue. And it's one of the more unusual connective tissues because it doesn't have what appears to be that important connective function. But I want to remind you that the characteristics of a connective tissue included a specialized cell, and we will see specialized cells in the blood. So the formed elements of the blood especially the erythrocytes, the red blood cells, and the leukocytes, the white blood cells. We'll see spellings for those in just a minute. They are the specialized cells. And then we have a third specialized cell, a megakaryocyte, that breaks off little cell fragments that are called platelets. So those are the three formed elements that are associated as the specialized cells. And then there's an extracellular matrix. And if you recall, the extracellular matrix is composed of a fluid or a gel, and it includes protein. And usually they're protein fibers. So together, this fluid or gel and the protein fibers make up what we talk about as the extracellular matrix. So we'll see in a moment what that extracellular matrix is and how it is that even though there are no fibrous proteins in circulating blood, it's still considered a connective tissue. So specialized cells include erythrocytes, the red blood cells, and their primary function is in the transport of oxygen and carbon dioxide. We hear a lot about the idea that red blood cells carry oxygen, but the same molecule that carries oxygen, hemoglobin, is also responsible, in part, for transporting carbon dioxide as the blood circulates. So erythrocytes are the red blood cells, leukocytes, and remember, leuko means white, erythro is red. So leukocytes are the white blood cells, and their job is to remove damaged tissue and to defend against invasion. So they've got two kind of roles. So if bacteria enter the body, then the leukocytes will go into action. Or if tissue gets damaged in the body and it's time to remove it, then the leukocytes can be assisting with that as well. Then the extracellular matrix is blood plasma. It's 92% water. And then the remaining 8% is primarily dissolved proteins. So these are not fibrous proteins, but these are proteins that are dissolved in the water that makes up blood plasma. And then I thought I'd kick in a couple of fun facts for you. The average blood volume of a human adult is about 5 liters, although that blood volume varies with body size, the electrolyte composition of the body fluids, of the blood especially, and the effect that having those extra salts or having fewer salts will have on fluid composition of the body. And then finally, the amount of adipose tissue, because people with larger amounts of adipose tissue are going to have larger amount of blood because there's a lot of blood vessels that are required to bring nutrients to those growing adipose tissue cells. Okay, so blood is connective tissue built of specialized cells and a specialized fluid with dissolved proteins, not fibrous proteins in it. And we got a fair bit of it, 5 liters. So when we go to describe blood, we can talk about different characteristics of it, including hematocrit, also known as the packed cell volume. So hematocrit, its abbreviation is HCT, packed cell volume, PCT. I think primarily the hematocrit is the term that you're going to hear out there in the medical field. And hematocrit tells you the percentage of the volume of blood that's made up by the red blood cells, by the erythrocytes. And normally, men have between 45 and 54 or so percent red blood cells. So that would be 45 to 54. The hematocrit is 45 to 54. In females, women, hematocrit is a little bit lower, 37 to 43% of blood volume is made up by red blood cells. And the reason there's a difference between men and women is because testosterone has a really stimulating effect upon a specific hormone that's produced by the kidneys. So when the kidneys notice that blood oxygen concentrations are getting a little low, then they will squirt out some erythropoietin, EPO, This is the same stuff that Lance Armstrong was apparently using to dope, and that many distance athletes, runners, and bicyclists and others who are competing in distance events, high energy use distance events. They dope with this because it stimulates the production of red blood cells. Erythro, again meaning red, poiesis is production, so it's a hormone that stimulates red blood cell production. So because men have testosterone, our kidneys tend to produce more erythropoietin than the kidneys of women. So if we were to fill a vial, like a centrifuge vial, with blood, say we fill it up to here, and then put it into a centrifuge and spin it round and round and round and round and round, that would cause all of the red blood cells to go to the bottom, toward the bottom of the tube, and it would separate the red blood cells from the blood plasma. And so these hematocrit values suggest that for women, slightly less than half, so about 40%, would be red blood cells. So these are women, and if we did it for men, spin that around, we would see, and here's the total volume, we would see little bit over half of blood volume is made up of red blood cells. Now, the remainder of the fluid that's left in this test tube after we spin, and send those heavy red blood cells down to the bottom of the tube would include a very thin layer. It's referred to as the buffy coat. A very thin layer, less than 1%, that is composed of the white blood cells and platelets that are found in the blood. There's not very much of that. And then the remainder, somewhere around 55%, again depending on whether you're male or female, would be the blood plasma. So here we have a man with, let's say, 47 or so percent hematocrit, and a woman with 40%. And so this leaves, and if the buffy coat is making up about 1%, then this would leave 52% of it being plasma. If we're looking at women, 59%, give or take, are going to have, or 59, I made a mess of that, sorry. Let's try that again. 59% of blood volume is going to be blood plasma. Okay. So there we are looking at the hematocrit, a way of describing the blood. So let's talk for a moment about hematopoiesis. Hematopoiesis is the process of making red blood cells, and all of the formed elements of the blood are built from, or are the result, they're offspring of these hematopoietic stem cells, sometimes referred to as hemo. Hemo means blood, cyto means cell, blasts are builders, so the blood cell building cells, or hematopoietic would mean, again, blood producing stem cells. And as these stem cells divide under the influence of erythropoietin, then the daughter cells will develop into red blood cells. There are other hormones that are going to control which blood cells these offspring cells are going to become. But as the stem cells divide off, they produce daughter cells that will not divide, well, that will then be influenced to divide again, and will give rise to one of many different kinds of potential formed elements. So what formed element that daughter cell becomes is all depending on these hematopoietic hematopoietic growth factors. Erythropoietin is one example of a hematopoietic growth factor, but and it's under the influence of these hormones then that the formed elements of red blood cells and the different types of white blood cells, or the megakaryocytes that are going to produce platelets, all develop. So exposure to EPO will cause production of more erythrocytes. Exposure to a hormone called thrombopoietin stimulates the development of the daughter cells into this thing called a megakaryocyte. And I said last night in class that mega, large, karyo, that's the nucleus. So it's a cell with a large nucleus. These megakaryocytes will then fragment, producing these small packets of cytoplasm inside of a bit of cell membrane that are the platelets. So thrombopoietin, thrombo is about platelets. Thrombopoietin will cause the production of more platelets in the blood. Okay, so hematopoiesis. So let's now talk specifically about the red blood cells. Red blood cells, I draw them like this with a little indentation in the middle. They are not a donut. There's not a hollow there. If we were to slice them in half like this and then look at the cut edge, what we'd see is this shape. So we talk about it as a biconcave disc. It's a flattened disc that has two concave surfaces on it. And the function of these concave surfaces are to increase the surface area of that red blood cell so that more gases can diffuse across the cell membrane, either oxygen coming in or oxygen leaving, depending on where the red blood cell is, or CO2 coming in and CO2 leaving, again, depending on the location of the red blood cell. So it places that cell membrane closer, by having this biconcave disc, places it closer to all the hemoglobin molecules that fill up the red blood cell. And it's actually those hemoglobin molecules. Hemoglobin is what makes red blood red. It's composed of two components. There's the heme molecule, and heme is a reddish pigment. It's the heme molecule that carries the oxygen. So each hemoglobin is able to carry 4 oxygen molecules. And then there's the globin portion of the hemoglobin, and that globin is a protein, and it is the portion of a hemoglobin that carries the carbon dioxide. So it brings those hemoglobin molecules here closer to the external environment. And if we're at the lungs, then we're going to bring in oxygen. We're going to load up those red blood cells with oxygen. And then when we get out to the body tissues, we're going to unload the oxygen from the red blood cells and dump it off in those tissues. Out there at the tissues, then we reload carbon dioxide. So the cells are producing waste carbon dioxide, and the hemoglobin molecule will pick that up. It'll bind to that globin portion, and once the red blood cell then returns to the lungs, then it'll dump off the carbon dioxide into the air of the lungs. So it's this process of loading and unloading depending on the location. And then the last thing that this biconcave disc shape allows is that the cell can flex some as it is making its way through the very narrow capillaries. So being able to flex is useful, and it's actually part of what brings a red blood cell's life to an end, is that it flexes so much that eventually it cracks and breaks and then gets removed from the body. So a red blood cell has a lifespan of only about 120 days, so that's something on the order of 4 months. By the end of those 4 months, it has accumulated enough damage that they can begin to leak. Oh, excuse me, I apologize for that yawn. Wow, I got to overcome. Sorry. So it, what was I saying? Yeah, so toward the end of those 4 months, then the red blood cell will crack a little bit or begin to leak, and then white blood cells will come along and digest them and get rid of those red blood cells. Okay, so that's a little bit of information. We've got a little bit more to talk about here. The cell membrane surface is studded with glycoproteins, identifier proteins. So if this is our, that was a terrible job, but if this is the biconcave disc, then hanging off of it are these glycoproteins that serve as identifier proteins. So they will. I'm sorry. Here I left my phone on. Man, I'm just breaking all the rules here, and I apologize for that. I can make this thing shut up. Okay, now I'm back again. So the cell membrane is studded with these proteins that hang off of it, and they serve as identifier proteins. They help to define the red blood cell as being a self cell if it's in the body. When they are mature, red blood cells don't have any nuclei. That's what anucleate means. It means that they have shed their nucleus. They break it down and get rid of it. And there's a couple of functions for this. Well, the primary function is that by getting rid of that big nucleus, it removes or it makes more space for packing that red blood cell with hemoglobin. But there's a consequence to not having a nucleus. Because there's no DNA inside a red blood cell, there's no ability to repair damaged red blood cells. And that's a big reason why they only last about 4 months in circulation. They also don't have any mitochondria. And this is important because you don't want the thing that's carrying your oxygen to be needing to use oxygen. They can still make ATP. They just do it through a pathway that doesn't require oxygen. It's called fermentation. So they can make some ATP from glucose, just not very much, not near as much as that glucose molecule has energy in it. All right, so no repairs, no ability to use oxygen. But they do make ATP. They do it. They just use a different pathway, one that doesn't require any oxygen to make it. So this is interesting. I guess I would have thought that I would have had this. Yeah, it looks like my slides got a little bit out of order. Yeah, no, didn't get out of order. So now let's talk specifically about erythropoiesis. Sorry, getting confused here. Erythropoiesis is the production of red blood cells, and it occurs in red bone marrow. So without red bone marrow, we're not going to be making any new red blood cells. And so we maintain homeostasis in hematocrit, in our red blood cell count, through a process known as negative feedback. I think you guys should know a lot about this now. So here's how the negative feedback system works. The kidneys and the liver are always paying attention to how much oxygen there is in the blood. And as oxygen gets low in the blood, then the livers and kidney the kidneys and liver, the kidneys and liver will release erythropoietin, and this erythropoietin then will make its way to the hematopoietic stem cells in the red bone marrow and result in increased production of red blood cells. Then as oxygen levels increase again in the blood, then the liver and the kidneys will reduce the amount of erythropoietin that they produce, and that will then allow the hematopoietic stem cells to slow down their production of red blood cells. So we have this constant push-pull of negative feedback that is going to control the amount of erythropoietin that's being released, that then goes on to control the number of red blood cells that are being produced. So under full stimulation by erythropoietin, so if you're getting your maximal EPO stimulation, then the body can make anywhere between 2 and 3 million red blood cells every second. That's just an astounding number to me. I cannot even picture how much that is. So it's easy to imagine then that it's possible for a condition called polycythemia to occur. That's excessive red blood cell production, and it can be dangerous because as you get more red blood cells, then there's going to be increased blood viscosity that slows down the flow of the blood. Another condition is anemia, and that's a deficiency in red blood cells. Or you may have the appropriate number of red blood cells, but each one will have less hemoglobin in it. And so it makes it more difficult, anemia does, to transport these the oxygen bound to the hemoglobin. All right. Last thing to talk about then is how do we get rid of that should not be on this slide. Sorry about that. How do we get rid of red blood cells? So at the end of their lifespan, as the red blood cells become damaged and because they can't be repaired, then the cell, especially the cell membrane, becomes very fragile and unable to bend or to flex, and as a result, the damaged cells will rupture as they pass through the very narrow capillaries of the spleen and the liver, and phagocytes will come along and will remove the cells and their cell contents. And the hemoglobin gets broken down into the 4 protein chains, the globin chains, and its associated heme group. That heme is further broken down into the iron that's contained in it, and this cool greenish pigment called biliverdin. And the iron is transported back to the bone marrow using a transport protein called transferrin. The biliverdin is further converted into an orange bile pigment that's called bilirubin. This is the reason why babies right after they're born tend to get real orange looking, is because they had a lot more red blood. They needed a lot of red blood cells when they were in utero because the processes of exchanging oxygen are more difficult. They happen more slowly at the placenta than they do in the lungs. And so when the baby is born and the lungs are now being used, got too many red blood cells, so we start taking out the red blood cells. Then we get all of this biliverdin that's being produced, and the biliverdin gets broken down into bilirubin, and now the child becomes orange looking. And so both biliverdin and bilirubin are excreted from the body as bile pigments, so they're part of the bile secretions that are coming from the liver, and we just pass them then with the feces. So that's good news. You can poop out these pigments either as bile pigments that are then released into the digestive system with the bile and pass out with the feces. And the globin molecule just gets broken down into amino acids, and then the globin molecules, proteins, can be rebuilt as new red blood cells are being made. So there you have it. Everything you wanted to know about red blood cells, but were probably afraid to ask. And I've given you some information about dietary factors and erythropoiesis. So if you'll just pay a little bit of attention to that, especially the forms of anemia that can occur, and the vitamin cofactors that really support the production of red blood cells. So we've talked a lot now about the functions of blood, about the formed elements in general, and then it's about the specific formed elements, the red blood cells. If you have questions, please don't hesitate to reach out and make contact with me, and I'll talk to you in the next lecture.